Hello dear students, welcome to the second class of excretory system. Today you will be learning about the second part of the structure of kidney. In the first session you have learned about the structure of urinary system, definitions of the terms used in the urinary system. You have learned the vertical or longitudinal section of kidney, etc. Today, in the class, you will be learning about the microscopic structure of kidney. That is, the structure and function of nephrons, which are the vital organs in the kidney for the formation of urine. Nephron is also called as the structural and functional unit of kidney. In each kidney, there are about over 1 million basic functional units. They are nephrons, where blood filtering and urine formation occur. Being a microscopic structure, the nephrons consist of two parts mainly: the renal corpuscles, which include the glomerular capillaries or glomerular tuft, which is also called as glomerulus of the nephron, and an encompassing capsule that is called Bowman's capsule. Altogether, that is, renal corpuscle is also called as Malpighian corpuscle. The second part, the renal tubule. This is the glomerulus, Bowman's capsule, and the tubules or renal tubules are the portion of the nephron which contains the tubules. The blood filtered in the Bowman's capsule enters or flows through the renal tubules for the formation of urine and then ends in the collecting duct system. Here is the location of the nephrons. Here you can see the location of nephrons in the cortical part of the kidney, and part of the nephron will be seen in the medulla part of the kidney. Each nephron length ranges from 45 to 65 millimeters. It has about 6 different parts. This is the nephron, the Bowman's capsule, which covers the glomerulus or blood capillaries. The tubular part is again divided into 4 distinct parts. The part which extends from the Bowman's capsule till the narrow bend of the tubulary part is called the proximal convoluted tubule. A hairpin bend-like structure which has 2 parts is called the Henle's loop or loop of Henle. The next part after the loop of Henle, here this again one more curve, is the distal convoluted tubule, and this part is the renal tubule, which opens into the collecting duct. This tubule is collecting tubule which opens into the collecting duct. This is the more precisely shown part of nephron, the blood vessels entering and leaving the Bowman's capsule, the capillary tuft here, the glomerulus, this tortuous part of the tubule, renal tubule, is PCT. Up till here, this is the Henle's loop, proximal convoluted, collecting tubule entering into the collecting duct. Let us learn in detail about the different parts of the nephron. The first part is Bowman's capsule. This Bowman's capsule acts just like a filter. Coffee is filtered in a filter. The Bowman's capsule, also called as glomerular capsule, renal corpuscular capsule, or capsular glomeruli, and also I have told you Malpighian corpuscle, is a double-walled cup-like structure that makes up upper part of nephron, the first part of the nephron. This capsule encloses a cluster of microscopic blood vessels. These are the blood vessels, which are also called as capillaries. Altogether, it is called as glomerulus, where wastes are filtered from the blood into the Bowman's capsule, which is continuous with the proximal convoluted tubule. This is the proximal convoluted tubule. The Bowman's capsule is divided into 3 layers. First layer or outer layer is parietal layer, which is made up of squamous epithelial cells. They have minute pores of diameter 12 nanometer. The second layer or middle layer is the middle basement membrane. This layer has a speciality that has selectively permeable capacity for certain substances. Next layer is the inner visceral layer, which consists of large nucleated muscle cells, which are called as podocytes. They possess finger-like projections called podoceles. They are also called as foot processes of podocytes. Podocytes are the cells in the Bowman's capsule in the kidney that cover around the capillaries of the glomerulus. I'll show you the podocytes and the layers of Bowman's capsule. The outer layer here is called the parietal cell layer. The inner layer is the visceral layer, which contain cuboidal epithelial cells. Squamous cuboidal epithelial cells, which contains the specialized muscle cells called podocytes. You can see here the podocyte. These are the blood vessels, small capillaries. And these podocytes, these bigger structures, are the podocytes, which contain small foot processes, finger-like things. They are called podoceles. This Bowman's capsule opens into the proximal convoluted tubule, which also contains epithelial cells, cuboidal epithelial cells. This enlarged picture shows you how it is present in the capillary wall. In small blood vessels, the walls of these blood vessels, the structure is like this. The first portion is the glycocalyx endothelial cells, glomerular basement membrane. The middle thing is the basement membrane. These have the specialized structure for the formation of filtrate, that is to filter the blood through this slit. It gets filtered through the glomerular basement membrane. These are the podocytes, which are bridged by the slit diaphragm, enters into the Bowman space here, or the urinary space. This structure is enlarged over here. Let us learn about the inner structure of the Bowman's capsule, which contains glomerulus, which is a network of small blood vessels or capillaries known as a tuft or glomerular tuft, located at the beginning of the nephron of the kidney. Capillaries of the glomerulus are lined by endothelial cells, as I've shown you in the last picture. These contain numerous pores called fenestrae. You can enlarge this diagram and see. These are the fenestration, small pores in the capillaries, glomerular capillaries. This is the afferent arteriole which carries the blood to the Bowman's capsule. Nodi, illi blood vessel enter agate. This same blood vessel gets further divided into numerous blood vessels, small blood capillaries. The structure of these capillaries are shown here. The fenestrations are present in the epithelial cell layer, and these are filtration slits in the glomerular basement membrane. The podocyte cells which are adhering to or covering the blood vessel. The foot process of these podocytes are the podoceles. This is the glomerular capillary. Here, the glomerular capillaries form a freely branching anastomotic network. Anastomotic andre connections irodu. Kondu connection irodu. Illinda afferent arteriole will carry the blood. The blood vessel itself gets divided into some about lobules, different lobules, about 6 lobules and each lobule of these consists of 3 to 6 capillary loops. Mattadu further divide agate. Altogether there will be about 20 to 40 loops in the glomerular tuft. The main function of this glomerulus or glomerular tuft is to filter the blood and release plasma only, here as filtrate, which passes down along the length of renal tubules. The first part of the renal tubule is the proximal convoluted tubule. After the Bowman's capsule here, this zigzag length or part of the renal tubule is called the proximal convoluted tubule, which lies between the Bowman's capsule and the next part, that is the Henle's loop. It is the major site for tubular reabsorption of water, nutrients, First, the Bowman's capsule will filter the blood and send it to the proximal convoluted tubule. Here in this tubule, the substances which are required by the blood will be reabsorbed, re-enter the bloodstream here in the proximal convoluted tubule. The proximal convoluted tubule has the simple cuboidal epithelial cells which are lining the inner side of the tubular region or lumen of PCT, that is proximal convoluted tubule. It has a brush border of microvilli lining the cuboidal epithelium of the PCT, which maximizes the absorptive surfaces during the formation of urine. The next part is the loop of Henle. This highlighted part is the loop of Henle. It's a U-shaped tube which consists of two limbs, the ascending limb and the descending limb. The ascending loop or limb, which consists of an initial short, thick portion and long, thin portion. ಇಲ್ಲಿ ಸ್ವಲ್ಪ portion, thick portions ಇದೆಯಲ್ಲ, ಇಲ್ಲಿಂದ ಸ್ವಲ್ಪ thick portions ಇದೆ, long, thin portion, and it has a hairpin bent structure and is continuous with the thin, long section or ascending limb of Henle's loop. Next part is the distal convoluted tubule. The distal convoluted tubule is lined with simple cuboidal epithelial cells that are shorter than those of the proximal convoluted tubule. The lumen appears larger in distal convoluted tubule than DCT. That to say, the DCT is broader than the proximal convoluted tubule. The point where DCT contacts the cortical thick ascending limb of the loop of Henle is called macula densa. It has tightly packed columnar cells. This picture, you can see, this is up till here, the Henle's loop extends from the proximal convoluted tubule. Here, this is the junction of Henle's loop and the distal convoluted tubule. The point at which, when it is in contact with the Bowman's capsule or near the afferent arteriole, in this part, it has specialized cells which are more sensitive to the changes in the concentration of the urine and blood pressure, changes in the blood pressure. So this part is called macula densa, and the cells here present in this part of the DCT is called macula densa cells. They are the tightly packed columnar cells. The next part is the juxtaglomerular apparatus. This I will take in the next class. Students, I have told you about the structure of NEPHRON, that is the structural and functional unit of the kidney. Since the time is exceeding, I will take up the next discussion about NEPHRON structure in the next class. Until then, happy learning. I'll be back with the third lesson, continuing with the structure of NEPHRON.